All right, so for our antibiotic sensitivity lab, we're going to set up the actual lab. So here I've got an agar plate. You can notice the agar in if you look at to the side. So this is the bottom of the plate. This is the top of the plate. So I've got my name and the organism that I'm going to be testing. So I'm going to be testing E. coli. So my E. coli actually is in a broth tube. I'm going to shake that up just a little bit to suspend some of that bacteria. So I'm going to need to transfer the bacteria to the plate, and then we're going to be testing 3 different antibiotics: tetracycline, chloramphenicol, and penicillin, to see which antibiotic is going to work on either inhibiting or killing our bacteria. So whenever we do this, we want to basically have this already labeled. We're going to need one of these sterile cotton tip swabs. That's this thing here. And then I also need my broth as well as my actual individual discs. So first thing I want to do is I want to actually put the bacteria onto the plate. So the way we're going to do this, we're going to flip the plate over, take the lid off, and then we're going to take this cotton tip swab here, and we're going to take a little bit of E. coli. So whenever I do this, I just want to make sure that I have my actual swab. I'm going to stick that swab into the into the broth here, and then I'm just going to squeeze some of that extra fluid off of that swab. I don't need a whole ton of it. So we're going to kind of do 3 angles to each other. So the first thing I'm going to do is I'm just going to gently move that, swab it back and forth kind of in a zigzag pattern. And then I'm going to do another angle here. Then I'm going to do a 3rd angle here. So I'm basically doing 3 angles to swab it. And then I'm also going to do the very edge of the plate too. So just around the edge. I'm not putting too much pressure. Once I'm done with that, I'm actually going to autoclave that, so that'll go into an autoclave bag. And so now we've got our bacteria on our plates. So now we want to actually put the 3 different antibiotics on them. So on each one of these, you'll notice I'll have them labeled. So this one's tetracycline. If you look very carefully, it's actually labeled TE30. So that one's tetracycline. Chloramphenicol is going to be labeled as C. It's also got C30 on it. And then penicillin here is this one. It's got P10 on it. So what we're going to do for each one of these is we're going to use a different set of forceps. These have all been autoclaved. So I'm going to use one set of forceps to place that penicillin disc. So I'm going to place it right on top of the agar that now has that bacteria on it. So I want to kind of place it and make sure that I've got space for all 3 discs, is basically what it comes down to here. And so when I place this, I want to place it kind of away from the edge. Once it drops, I don't want to touch it too much. I'm just going to press it in with the tips of those forceps, and then I'm going to move on to the next set with a different set of tongs. So here I've got another set. This is my tetracycline. So I'm going to grab the tetracycline disc and do the same exact thing. I want to make sure that these discs are in a spot that they're not on top of each other. There's enough space around to create that zone of inhibition, and also just to make sure that they're not very close to the edge as well. So that one dropped, just going to kind of press it in. And then the last one is going to be the chloramphenicol here. So same thing, I'm just going to grab the disc, place it on the agar, kind of gently push it in just a little bit, not too tough, and then place the lid on top. So when we incubate these, we're going to incubate them upside down. And so the reason for pressing that on is so that way we can incubate them upside down, and we're going to put them in the incubator. Once we're done, we're going to let it incubate 24 to 48 hours. After that, we're going to measure the zone of inhibition. The zone of inhibition, we're going to have a lot of bacteria that we swabbed on here, and hopefully those antibiotic discs are actually either going to inhibit, in which case it's a microbiostatic, or if it kills, it's a microbicidal. And then we're going to be able to measure that clear area around the actual discs. And so we'll have some pictures of results on that as well. So once again, 3 different antibiotics: chloramphenicol here in C, P is for penicillin, TE is for tetracycline. Incubate it for 24 to 48 hours, closer to 24. After that, if we aren't able to look at the results right away, stick it in the fridge. And then once we're actually back in class, we can look at the results and measure those zones of inhibition.